All right, good morning, everybody. My pleasure to welcome you to what is going to be a really great talk.
Oh man, that's a lot of pressure.
I'd like to welcome.
Good to great. Good to great.
I'd like to welcome Dr. Sliman Bensmaia to Case Western Reserve University. Dr. Bensmaia did his PhD at the highly esteemed University of North Carolina at Chapel Hill. That's an inside joke.
There's no need to cackle about that.
He did a postdoc at Johns Hopkins University before becoming faculty at the University of Chicago. He's gotten a number of awards, including a number of early career awards from the National Science Foundation, IEEE, a number of distinguished researcher awards. The best thing, though, is that he does really, really cool work, which he's going to explain to us today, really focusing on sensory encoding and intracortical microstimulation. So, without further ado, welcome Dr. Bensmaia.
Sweet.
So first of all, this is like one of the, if not the, earliest talk I've ever had to give. And I feel like you guys hate yourselves for having to do this once a month. But, you know, I only have to do this once, so it's all good. So before I get started, I just want to tell you a little bit about what we do, what we spend most of our time doing in my lab, just to sort of frame everything else that I'll be talking about. And what we spend, you know, what 3/4, probably 4/5 of my lab spends, you know, does, falls under the rubric neural coding. And we're going to talk about touch, so I'll give you a touch example. So when you grasp an object, it results in a spatiotemporal pattern of deformation of the skin, produces a pattern of activation in the nerve, and these different colors, you know, this is cartoon, but these different colors correspond to different types of receptors that innervate the skin and send different types of signals to the brain. And then these patterns of activation of the nerve culminate in patterns of activation in the brain, first in the cuneate nucleus, then in the thalamus, and then various stages of processing in cortex, ultimately culminating in a percept of this object. And when you grasp an object, you have all this information about this object coming from your hands, information about its size, its shape, its texture, its motion across the skin. And somehow all these patterns of activation, the sensory information is contained in these patterns of activation. And what we spend most of our time doing is what aspect of the nerve activity or the brain activity conveys information about texture, what aspect conveys information about shape, about motion, etc. And we come up with models where we can actually say, you give me a pattern of neural activation and I can tell you a lot about what the thing was that produced that pattern, right? So if you give me two patterns, I can tell you which one was the rougher one, or how distinguishable were their textures, or if it had a shape, what was the shape, right? So that's what neural coding is. That's not what I'm going to talk about today, though. But we're going to try to leverage what we've learned about how the intact nervous system encodes this information to try to restore that information in different populations of subjects. So you guys are super familiar with this, right? So with amputees, they've lost their sense of touch because they've lost their arm. And the idea, you guys have some of the, you know, if not the best, some of the best stuff on this, which is to replace the hand. And for reasons I'll show you in a minute, and you probably already know, it's not only important to replace the hand with a bionic hand and being able to move it volitionally, but you need to restore these sensory signals. And you can do that by interfacing with the nerve. And the idea is, like I said, all these sensory signals coming from the hand are carried through the nerve. And if you can stimulate the nerve to simulate these afferent responses, you can, in principle, restore the sense of touch in all its glory. That's not what we're going to talk about today. I'm going to talk about a second population of patients, tetraplegic patients, who have typically an upper spinal cord injury and who are insensate and paralyzed from the neck down. And for those patients, stimulating the nerve doesn't help because the nerve is no longer attached to the brain. So it doesn't result, or in many of them, it doesn't result in any kind of sensation. So for these patients, we have to interface directly with the brain. So, but before we go into that and our efforts in trying to restore touch through a brain interface, we need to talk, and maybe because you guys are a case, you guys already know this, but I'm just going to spend the next few minutes trying to convince you that it's critical to restore these sensory signals. This is a movie I made of me doing stuff with my hands. Most of it's super mundane stuff, like picking things up, writing with a utensil, typing on a keyboard, etc. All these things are things you could not do without sensory signals from your hands, because anytime you're interacting with an object, you get this barrage of sensory signals that tell you about the object and about contact events with the object. And without these sensory signals, basically your hands would be useless. Let me prove that to you. This is a movie that a colleague of mine, Roland Johansson, made of a lady lighting a match, no problem. Same lady, moments later, she's sober, she's sighted, she's neurologically intact, she's gonna do the same task. The only difference is her fingertips have been numbed. She drops that, she tries to pick it up. She can see the thing, but she's like, man, there's no way I'm gonna pick that up. And then finally, after a long struggle, and you can tell she's doing it in a weird way, right? But after a long struggle, she manages to light this match. And she's gonna be doing this over and over and over again, and she's gonna continue to suck at it. She never gets any better at it. And that's because the visual system is does not convey the kind of information you need to do this kind of thing. To do this kind of thing, you need your sense of touch. This next guy suffers from a peripheral neuropathy that has robbed him of all somatosensory signals coming from his entire body. And he's doing something even simpler than lighting a match. He's just trying to grab a coffee cup and bring it to his mouth. And if I showed you this guy, and you didn't know anything about him, and I didn't tell you anything about him, and I asked you, what do you think is wrong with him? You would think that he has a severe motor impairment. And that beautifully illustrates the link between the somatosensory and motor system, the yin and the yang between the somatosensory and motor systems, where one does not work without the other. So that's true of, if I take away your somatosensation, suddenly this is what you're like, right? Intact motor system, this is what you're like. Now, in the context of a brain control prosthesis, so this is a movie that was made by my colleagues at the University of Pittsburgh. This is Jen here. Maybe a lot of you have already seen this, but this is Jen here. She's tetraplegic. She's been implanted with an array of electrodes in the primary motor cortex. This is the part of the brain that sends signals to the muscles to move the arm. And my colleagues at Pitt, and there's a cottage industry of people doing stuff like that, have devised algorithms to read these signals from primary motor cortex and infer the intended movements. The idea is when you move your arm one way, there's one pattern of activation. You move your arm another way, there's another pattern of activation. And they can look at these patterns of activation and infer what the subject wants to do, or the patient wants to do, and can move the robotic arm, right? And I'm not gonna spend a lot of time talking about that because that's not the focus of my talk. But the point is, if she volitionally tries to move her arm in a specific way, there goes the robot, right? And so she can control this robot by thought, and if you're seeing this for the first time, which I doubt any of you are, you're like, wow, that's like an amazing scientific and technological achievement. This person can control this extracorporeal device with her mind and can do so pretty adroitly. But if you see this for like the 1000th time, you realize that you would be way better at this than she is. You'd be like, woof, woof, woof. But she's about as good as you would be without a somatosensory system. And so the idea is, if you want to restore the kind of dexterity that we are endowed with, you need not only to have these pretty sweet anthropomorphic robotic arms and control systems to actually move these arms volitionally, but you also need to restore the sensory system, and that's where we come in. This is for fun. This is not a good movie, but I like the scene of somebody playing guitar with two prosthetic hands. The point is, you're not going to play guitar with a prosthesis without the sense of touch. Fact. So how do we go about this? This is a little movie that was made to sort of illustrate this. The idea is, anytime you interact with an object, you have a skin deformation, produces a pattern of activation in the receptors in your skin that depend on what you touch and how you touch it, ultimately culminating in a pattern of activation in somatosensory cortex. So this is the part of the brain that receives signals from the hand. And those patterns of activation depend on what you touch and how you touch it. Now, these prosthetic hands have sensors in them that replace the native receptors. And what we're trying to do is come up with algorithms to take the output of these sensors and convert them into patterns of electrical stimulation of the brain to reproduce these natural patterns of activation. And remember, these natural patterns of activation in the brain is what we spend most of our time doing, right? Or studying. We spend most of our time trying to figure out how is it that these patterns of activation sort of convey information about the external world. So that's the idea, is to create an encoding algorithm. So this project, that I'm going to talk about today was part of Revolutionizing Prosthetics, which was a DARPA-funded project, and I want to mention that because, as probably a lot of you know, unlike other funding agencies, where they might give you some money and say, hey, discover something cool or something. That's not the way they roll at DARPA. At DARPA, it's like, you have 3 years to do 10 years worth of work, and we need this. This is the thing that we need. And there's deliverables and specific deliverables. And so we couldn't just set out and say, we're just going to restore the sense of touch, right? We had to kind of make this a tractable problem. I mean, we're still trying to figure out how the sense of touch works. That's why I probably have a long career ahead of me still to try to figure that out. But we wanted to restore key information about contact events. And we thought, okay, what is the basic information that you need to grasp an object, right? Let's start with that. What is the basic tactile information you need to grasp an object? So minimally, you need to know that you're touching it. You need to know where you're touching it. So the thumb and one of the fingers has to be touching it in order to be able to grasp it. And you need to know how much force you're exerting on the object so to be able to pick it up without crushing it or just exerting unnecessary amounts of force on it. And all of those things are, I mean, the primary source of information about those contact events comes from the sense of touch. For instance, we're exquisitely sensitive to how much pressure we exert on something, and we exert just enough pressure so as to not drop it and not more than that, right? Without the sense of touch, we just crush everything or drop everything. So contact location and contact pressure were the two kind of streams of information we wanted to convey, and we wanted to do so using a biomimetic approach. And there's different schools of thought. The biomimetic approach, in a way, is sort of the simple-minded way, right? You're like, okay, well, in intact individuals, this is how information is encoded. Let's do that, right? Because it works for intact individuals, so it should work for tetraplegic patients. There's another school of thought, and I just want to spend 3 minutes talking about this because I completely disagree with this other school of thought. It's very, it might piss some people off, but so be it. The other school of thought is that the brain is this plastic learning device, right? So if you create a mapping between the contact events and patterns of electrical stimulation of the brain that is systematic, it can be arbitrary, but as long as it's systematic, the brain will figure out how it works, right? So one of the kind of famous proponents of this view, like one of the extreme proponents of this view, is Miguel Nicolelis. In principle, it makes sense. Maybe that can work. But think about how, and that can work under certain circumstances. People have shown that these kinds of arbitrary mappings can be learned after many, many tedious hours by both humans and animals. However, if you think about, usually it's like two-dimensional. Like you can learn to associate the cursor moving up with one, patterns of stimulation and cursor moving right with another pattern of stimulation. But when you think about hands and arms, they are so complex. There's like 30 degrees of freedom of movement. So in order to track the hand, you need basically 30 dimensions of feedback. And then every contact event, the tactile space is hugely dimensional, but let's say at least dozens of dimensional of tactile space. And so to try in adulthood to learn such an incredibly complex space, where basically this random tingling sensation I experience when I move my thumb, this other tingling sensation I experience when my index finger touches something, having to learn all that stuff in adulthood, I don't think is possible. No one has demonstrated anything like that, and so it's a challenge that some people are working on, but I don't think will ever be overcome. In fact, shameless plug, we wrote a paper about this in Trends in Cognitive Science last year, looking at the literature on plasticity, and I think showing that there's nothing in the literature on plasticity, especially post-deafferentation, that would suggest that we can learn these completely new sensory representations in adulthood. But I welcome any challenges to that idea later, and I'm not even dogmatic about it. I'm just throwing down the gauntlet and say I don't think you can do it. And so, if you can't, either way, even if you can do it, conceptually it makes sense to the extent that you can actually reproduce natural patterns of activation. That seems like the way to go because people won't have to learn anything. If you can reproduce them exactly, then it's like touch is back, right? So that's the sort of motivating sort of framework to what I'll be talking about. Okay, this is a sort of drawing of somatosensory cortex. I'm sure everyone knows this already, but you have a complete body map in somatosensory cortex. Again, somatosensory cortex is the part of the brain that receives, the part of the cortex that receives all, the signals from the body, so proprioceptive and cutaneous signals. You have a complete body map, which means, the legs are down here, the hand is here, the face is here, the arm is here. And even within the hand, actually, you really have 4 body maps. One in Brodmann's area 3A, one in Brodmann's area 3B, one in 1 and 1 and 2. And even within the hand representation, you have this very systematic organization of the digits where the thumb is anterior and lateral to the index, is anterior and lateral to the middle finger, etc. So you have this beautiful hand map. And that's where we stick our electrodes. And by the way, the work I'm going to talk about is first from macaques, rhesus macaques, which is basically the sort of more human-like primate you can stick electrodes into. And then at the end, I'll talk to you about how we can leverage what we've done in macaques and humans and have leveraged what we've done in macaques and humans. So the array technology that we're using, we use 2 types of technologies. One is the sort of the industry standard Utah electrode array that I'm sure a lot of you have seen before. It's 100 electrodes arrayed over a 10 by 10 grid, 4 by 4 millimeters. And that's it's nice because it's approved for human use, and that's what all the human patients I'll show you have been implanted with. And the problem, though, is there's all kinds of problems with it. But one of the problems is the fact that the electrodes are a maximum of 1.5 millimeters long. And some of these sensory representations that we need are deeper than that in the bank of the anterior bank of the central sulcus. And so we needed longer electrodes. For that, we use floating microelectrode arrays. So we stick these electrodes in the brain. We wait a week, and then we can map we can recreate these brain maps. So for instance, this corresponds to this UEA here. And so these electrodes here respond to the palm, these electrodes here respond to the middle finger, these electrodes here respond to the index finger, right? So this is sort of a first pass. I'm going to show you this 2 more times. This is the first pass at showing you the somatotopic organization and how we can reconstruct it. By the way, if you have any questions, feel free to ask. Okay, so that's our neural interface. Now, if this were a human, if we could do this in humans, right, this would be in some ways easier. In some ways it would be way harder, but in some ways it'd be easier because humans have speech. So you could stimulate and say, hey, what did you feel? And where did you feel it? And they'd tell you what and where, and you would get a Nature paper. The problem, though, is that they won't let you do that in America or in most of the world. They won't let you stick electrodes until you prove that it actually does something productive. So we had to start with experiments with monkeys, and the issue with perception is that it's an internal thing, right? And so we had to somehow probe this internal thing in a non-verbal creature. And so how do you go about that? Well, the strategy we adopted was to train the animals to discriminate pokes applied to the hand. And once the animals, and I'll describe in more detail what the specific paradigms were. And once the animals had learned to discriminate these pokes, we started replacing one of these pokes with an electrical stimulus applied to the brain. And we wanted to see if the animal behaved as if it had been poked a specific way. And if it behaved as if it had been poked a specific way, we could infer something about what they'd felt. And that'll become clearer in a minute. So because you guys are, neural engineering nerds, or a lot of you are, I'm going to tell you some of the details that I kind of gloss over. Most of the electrical stimuli were biphasic symmetrical pulses. Unless otherwise specified, you had 200 microsecond phases at 300 hertz. But as you'll see later, we kind of varied those parameters also. So those were the electrical stimuli. They were delivered using BlackRock gear. And then the mechanical stimuli, we designed this robot that allows us to very precisely indent the skin wherever we want with microns precision. And of course, all the tactile stimuli were completely innocuous. Okay, so I told you we were trying to convey 2 types of information. The first was information about contact location. Where is it that you touch things, right? How does your brain keep track of where your hand is touching stuff? So And we wanted to first think about how is it that your brain keeps track of that, and then try to reproduce that through a neural interface. So this is a sort of a more sciencey version of what I showed you earlier. So this is, if you use our little robot to poke the second palmar whorl, this is the pattern of activation that's evoked over the entire array. So this is the 4x4 millimeters of cortex. Blue means not active. Red means very active, and these other intermediate colors mean intermediate levels of activation. And so you can see that when you poke here, you get activation that peaks here, right? Now if you poke the third palmar whorl, you get activation that peaks here, and you poke the pinky, you get activation that peaks here. So this is what I told you earlier. You have this map of the hand, systematically organized map of the hand, where if you poke different parts of the hand, you get patterns of activation in different parts of the brain. So then the question was, and this is pretty straightforward, and that's why I start with this. We wanted to see if you stimulate an electrode that, say, responds to the index finger, will the animal have a sensation that is referred to the, or will the animal feel something on its index finger, essentially, right? So how do we go about trying to probe where the animal feels the stimulus? Well, we trained them to do a mechanical discrimination task, where we would poke them with the robot on, say, the ring finger of the left hand, and then on the middle finger of the left hand. And the animal's task was to say, was the second poke to the right or the left of the first, right? So in this case, it was to the left. And it takes 6 months or like maybe even a year to train these animals to do this task so that we can poke them anywhere on the hand, and they learn to generalize it. They finally figure out what it is that we're asking them to do. It takes forever. Once they've plateaued and they stop getting better at it, and they get pretty good at it, I'll show you the data in a minute. We started doing this. We started interleaving these mechanical trials with these kinds of trials, where we might touch, the middle finger, and then electrically stimulate through an electrode that we have previously determined responds to the ring finger. And then if we're right, the animal should respond right? If we're correct, the animals should respond right. And here's an important design. So you could teach an animal to respond right here. That would be trivially easy. It would take like an hour or maybe less. Any kind of pairing of stimulus where you poke them on a place, and then you give them any kind of other stimulus, and then you reward one response over the other, they'll learn to do that, right? So we want to make sure that they're not just learning arbitrary stimulus response pairings. And to do that, on any given experimental block, there were many, many different conditions. So we might poke them in different parts of the skin. We might electrically stimulate them through different electrodes, so that they had no opportunity to learn what the right answer was based on these arbitrary kind of pairings. They had to discriminate the perceived location of either the mechanical pokes or mechanical and electrical stimuli. And so we would do one block with one set of conditions, and then the next block would be a different set of conditions. So they couldn't even transfer their knowledge or their experience from block to block. This was to ensure that the animals had to do this based on perceived location. So this is what the data look like. So this is the performance of the animals when both stimuli were mechanical. So we poked the animal, said, was the second poke to the left or right or the first? And so you can see, on average, the animals are around 90%. Sometimes, and each of these points corresponds to a pair of locations. Sometimes they're perfect when the pokes are really far away. Sometimes they're not very good when the pokes are close together. And then you have a spread of performance. But overall, it's about 90%. This is the performance when we replace one of the pokes with an electrical stimulus applied to area 3B, right? So that's really primary somatosensory cortex proper. This is the station in the brain that first receives input, or the cortex that first receives input from the hand. This is sort of a second area, like a higher level area. So each of these points was sort of associated with one of these locations. So we always, anytime we picked a receptive, or for every receptive field that we got in 3B, we would pair it with a mechanical stimulus there. And so you can see that the performance when one of the mechanical stimuli was replaced with an electrical stimulus applied to 3B was nearly as good. Actually, because this is paired, there is actually a significant difference between this and that, but it's, numerically a tiny, tiny difference. So the animals were almost as good when we replaced one of these mechanical stimuli with an electrical stimulus applied to area 3B. They were worse when we placed it with an electrical stimulus applied to area 1. And if you're a somatosensory nerd, this makes a lot of sense. Area 3B is primary somatosensory cortex proper, has tiny receptive fields compared to area 1, which sometimes have receptive fields that span a couple fingers. So it's not surprising that the projection fields would be smaller in area 3B than area 1. And so this is consistent with that. Sure.
What about the spacing of the electrodes?
Right. So we didn't look at the acuity, like how close, can the animal distinguish the electrical stimulus applied to an electrode to an electrical stimulus applied to an adjacent electrode. And the reason why we didn't is because we didn't know what the ground truth would be there. When both stimuli are electrical, like, can you distinguish things that are close together or not? And if we start giving the animal trials where they just don't feel the difference, then you're just going to confuse them and everything is going to, they're not going to be able to do this task anymore. So that's not something we can do on animals. But so what I can tell you though, so I can't tell you what the spatial acuity of this task is. And I will argue that in the current state it doesn't matter very much. But what I can tell you is that they could distinguish a stimulus on the palmar whorl, on this palmar whorl, or this palmar whorl on this fingertip versus that fingertip, or even this fingertip from that fingertip. So we don't know how acute this perception is, but we do know that it's. So let me, in fact, get to the punchline here, that if you electrically stimulate through individual electrodes, penetrating electrodes in somatosensory cortex, you get these sensations that are referred to a relatively small patch of skin.
I just have a real quick question methodologically. You had a mechanical device moving around or electrical. Did you move the mechanical the same way, whether it was electrical or mechanical? And then was the animal...
well, so the animal couldn't see anything, right? We use white noise to mask all sounds. Anyways, the other thing is that when we did this, I'm going to convince you in a second that this worked because we have human data now. But no, fair enough. The other important thing is that when we did this task, this is key because, well, I'll tell you why this is key in a minute. We trained the animal to do the mechanical task, and they got to 90% or something, and then after a few weeks, they stopped getting better, so we're just like, okay, this is the plateau. The day we do the electrical task, they get to a performance with 3B that's nearly as good, and they never improve, right? They never got any better. So, so that means that they really just generalized the mechanical to electrical. And I want to bring this up because for those of you who are familiar with Rodolfo Romo's work, you get the impression reading, and this is the frequency discrimination where the animal, we're doing tactile frequency discrimination of vibrations applied to the skin, and then his team replaced mechanical with electrical stimuli. And if you read the papers, you get the impression that they just transferred immediately. They did not transfer immediately. They had to basically relearn the task completely when in the electrical case, right? So in this case, they didn't, which I think is important, allows us to kind of infer that this is a spatial discrimination task and not some other task that they,
Yeah, so Bob was asking about the spatial resolution in the cortex, but did you measure if the tactile or the mechanical spatial resolution was equivalent to spatial resolution when you were doing mechanical versus electrical?
No, we didn't look at resolution at all. But let's just think about resolution a little bit for a second. The other reason why resolution is not super interested in monkeys is because their brains are way, way smaller than the human brain, and if the array is the same size, then your resolution should basically get 10 times better in humans in principle. But a fingertip representation is roughly 1 millimeter wide, right? So you basically have two electrodes per fingertip representations given this technology. So best case scenario, you have you can tell this side from that side. That's the best you could ever do. We never, but because there's probably some spread, again, in monkeys we can't really look at that because there is some, so with the mechanical we could have, because in the mechanical we could bring them arbitrarily close and we would know what their relative spatial position is. But when you're mapping receptive fields, you know, you say, okay, it's this area, and if you're doing, it's this area versus this area, they're overlapping. There's no right answer, right? That's why we wanted to make sure there was always a correct answer when you were looking at electrically induced sensations, so as to not confuse the animals. It's hard enough, trust me. It took them a year to understand that. We don't want to confuse them unnecessarily. And this was just sort of a basis of, can we get spatially localized percepts? And then when you get to the humans, then you can actually do these kinds of experiments.
And then it's sort of related, so then did you do the similar study where you just had purely electrical stimulation without the mechanical?
You know what? We never did that. We should have, but we never did. Where we had 2 electrical stimulation. But you know what? It all worked out in the end. Whatever skepticism you have, keep it bundled up for another 20 minutes. And then you're just gonna open that and just dump it out. Okay, so the conclusion from this experiment, one that I will come back to later when we talk about human studies, was that electrical stimulation through individual electrodes in somatosensory cortex elicits very spatially localized percepts, localized to a finger pad or palmar world. And later I'll tell you how that we can use that to convey information about contact location in a very sort of intuitive way. So the other dimension that we want to look at was contact pressure. So the idea is, you know, we exert very precise amounts of pressure on objects, just enough to grasp them and not drop them, right? How is it that our brain tracks that information? Notice that I always take a sip when and this is an excuse every time I talk about grasp to have a sip of coffee. So the sensory correlate, when you touch something lightly, you get this faint sensation. When you touch something, you know, or something is jammed across your skin strongly, you get the strong sensation. What is the neural correlate of that? So this is our recordings from primary somatosensory cortex when we indent the fingertip. This is the pinky fingertip. If you indent it lightly, you get this weak, very localized response. And as you increase the pressure exerted on the skin, what you see is that these guys here become more and more active. And this is like a plot showing that. This is the firing rate as a function of amplitude. This is exactly what you would expect, right? If you know anything about neuroscience, this is what you expect, that the firing rate increases as a function of amplitude. But the other thing that happens is that you have recruitment, like there's more and more neurons that get activated as you increase the pressure. As shown here, this is the surface of cortex that is significantly above some kind of threshold of activation. So we reason this is the code in the brain for pressure, right? You want to increase the activation of neurons that are right there in the center of the receptive zone, which we've called the hot zone, and you want to mimic this sort of progressive recruitment with increases in pressure. And the nice thing is, you can sort of oh yeah, go ahead.
So are you talking about second somatosensory area over here too, right?
This is, okay, you know what? This is the second talk in a row where somebody reminds me that I need to change the slide. We thought, it turns out that all of this is area 1. This is an early slide. All of this is area 1. We thought that this was between area, that we had a little bit of area 1 and area 2. I need to make a note, and I'm going to forget all about this, and somebody else is going to embarrass me next time I give this talk. But we thought that there was a separation because you know how there's like this mirror here, and if you look across all our skin locations, it seemed like there was a hot spot here and a hot spot there. A point of activation here, and we thought it corresponded to 2 areas, area 1 and area 2. But then we consulted with John Koss, and he basically told us that area 1 is at least 4 millimeters wide, right? And because we're hugging the central sulcus, it's very unlikely that there's any area 2 at all over this area, over this electrode. So basically, all of this is area 1.
Can I ask you a question about the standardized firing rate mean?
Yeah, sure. So The reason why we're doing standardized firing rate here is because if you want to combine across, so this is with, actually it doesn't even matter. If you want to combine firing rates across a bunch of different electrodes, right? And these are multi-unit recordings, right? So if you want to combine responses, let's say on electrode 5, you have 3 units, and on electrode 10, you have 20 units. I mean, these are numbers I'm making up. Then you have to sort of put these on the same scale, right? So that, you know, if you're going to average across to show what the trend is across all electrodes. So what we did here, and this is true of all the data I'm going to show you, all the neural data I'm going to show you, is we took the baseline activation and computed the mean and standard deviation of that baseline activation. And then we subtracted from all activations, we subtracted that mean and divided by the standard deviation. And if you don't do that, right, if you don't, you just report the firing rates, like on these maps, if you report the firing rates as they're recorded, basically what you're going to see is, what you're going to get back is not these nicely spatially patterned of activation. You're going to get this mess because some electrodes are highly spontaneously active and really kind of have high slopes. Others have narrower slopes. And so you have to standardize for sort of basically the number of units and their baseline activity. That's what we're doing here across the board. But if I showed you, to maybe assuage some concerns you might have, if I showed you the response of individual electrodes, they would all go like this, right? This is just to combine them, we basically put them on this standardized metric, which is subtract mean divided by standard deviation.
So saying 2 means 2 hertz.
Oh, I'm sorry, yeah. So 2 means 2 standard deviations above the mean of the baseline mean.
So in the normal response, if you center surround where the surround areas might get inhibited, are you able to reproduce that with the electrical stimulation?
So I think what you're referring to is if you look at the receptive field of a neuron in area 3B, you have excitatory zones flanked by inhibitory ones, right? But if you look at the aggregate activity of neurons, because of their sort of idiosyncratic organization of excitatory and inhibitory zones, you don't see that. You just see basically, you know, it's all excitatory basically. Because remember, all these neurons with, you know, this is population activity, aggregate activity of hundreds or thousands of neurons, right? So you don't see the receptive field structure of individual ones. Overall, all this results is in excitation.
You never see the decrease in spontaneous rate in the flanking?
Not in aggregate activity, no. Okay, so anyways, let me just get back on track here. So the point of this slide is that 2 things happen when you increase pressure. You have an increase in the activation of these guys here in the center, and you have progressive recruitment of neurons around them. And this is systematic across arrays, across locations, across monkeys. And so we hypothesized that is the key sort of neural code for pressure, so we wanted to reproduce that, and it turns out you can do that just by increasing the juice. Just increasing the current is going to do both of those things. It's going to result in more, in exciting neurons near the electrode tip more, and is going to result in current spread away from the electrode, thereby recruiting more neurons. So now we need to probe the animal's perception of pressure, right? So this is a completely different sensory dimension that we need to then kind of be able to probe. And so we had to train these same animals to do a completely different task. In fact, we trained them to do 2 tasks. And these are ultra standard psychophysical tasks. This is how you would measure it in a human, right? Where you have two intervals that are, you know, that are indicated by lights on the monitor. Actually, the structure is identical to the one to the location discrimination task, where you have 2 intervals. But instead of the 2 pokes being in different locations, now they're in the same location. Well, in this case, there's only 1 poke. And you have a stimulus that's either a mechanical indentation that's either presented in the first interval or in the second interval. And these are really small indentations from a 1/20th of a millimeter to like a 1/3 of a millimeter. And the animal's task is to say which of these 2 intervals contain the stimulus, right? So this is a standard detection task. And here we're probing how faint a stimulus can the animal reliably detect. Was there a question? Did I just hallucinate? That was weird. Okay. The other task which we trained them to do was a pressure discrimination task. So now there are two stimuli, and the animal's task is to say which of the two were stronger. And in these tasks, there's one stimulus that's the same on each trial. It's called the standard stimulus. In this case, it's in the second interval. In this case, in the first. And it's paired with a comparison stimulus that's variable intensity, varying from, in this case, 300 microns to 2 millimeters. And then we had a high standard at 2 millimeters that was paired with comparisons that varied between 150 microns and 1 millimeter. Bear with me. This is some kind of nerdy psychophysical stuff. We'll get back to the encoding algorithms in a minute. The important design feature here is that all these trials were interleaved, so the animal really had to pay attention to both intervals to know what the right answer was. And this is what the data look like. So for detection, as the amplitude of the stimulus increases, so now there's one stimulus per trial, and the animal is supposed to say which of the two intervals the stimulus occurred in. Obviously, as expected, as the amplitude of the stimulus gets stronger, the animal can perceive it better, and therefore can more reliably identify which interval it occurs in. And that's why you get the psychometric function that increases. And you can define a threshold as what is the intensity of the stimulus that you need, or the amplitude of the stimulus that you need for the animal to detect it 75% of the time. And in this case, it was 140 microns. So that's pretty small. For mechanical pressure discrimination, it's the same thing here, basically the same thing. So for the 150 micron standard, that means one of the stimuli is always 150 microns. The other one varies from 300 to 1 millimeter, right? And as the other one increases in amplitude, it becomes more and more different than the 150 micron stimulus, and therefore it's easier to detect it as being more intense, and that's why this goes up. The reason why this goes down is because the comparison now is 2,000 microns. As the comparison stimulus increases, it becomes more and more similar to the 2,000 micron stimulus, and therefore that's why performance goes down, right? And here you can actually define JNDs as how much do I need to change the stimulus for the animal to detect that change 75% of the time. And so the JNDs are huge, like 0.5 millimeter and 1 millimeter here. Note, by the way, that, and this is Weber's law, this is a well-known perceptual law, that you need more of a change for you need a smaller change for the small stimulus than for the large stimulus, right? This is in all perceptual, natural perceptual modalities. You see this kind of thing, where the JND, the just noticeable difference, increases with the standard amplitude. It's going to be relevant in a minute because that's not true of electrical stimulation. So then, okay, so we trained these animals to do this task. They plateaued in their performance of the task. We got the mechanical data, and then we did the exact same thing with electrical stimuli applied to the brain. So now in the detection task, there are two intervals. An electrical stimulus is presented during one of them, and just like we saw with the mechanical, the performance improves as the electrical stimulus increases, just as you would expect. Then we went in a little cray-cray, and we, this is a new sensory modality, right? There's parameters. Frequency, amplitude, pulse width, duration. When you're studying a new sensory modality, you need to understand how all these parameters affect the perception. So we did this in tedious detail. So first we looked at the effect of pulse width on detectability, and we found that pulse width, as expected, detectability improves with pulse width up to a point. As you know, and that makes sense because you're just injecting more current as you increase the pulse width. We looked at the effect of frequency. Interestingly here, the frequency, the JNDs gets lower and lower, so sensitivity improves as a function of frequency up to a point, and then it stops improving at around 250 hertz or so. Duration, as you increase the duration of the stimulus, the animal gets better at detecting it up to a point, and then stops, like after about 100 or 200 milliseconds. You can make the stimulus longer, and if the animal doesn't detect it at 200, it won't detect it at 1 second. And then there's a confound between frequency and duration. Maybe the key is how many pulses there are, right? It turns out it's not just how many pulses there are. Frequency and duration matter independently. So this is the detection. We did kind of a bunch of stuff with discrimination too. So this is with the low standard, you have this upward going function, a low standard of 30 microamps. With a high standard, you have this downward looking function. And here's a fun fact. The JNDs don't change significantly as a function of standard amplitude. So this is a fundamental difference between mechanical stimulation and electrical stimulation, where the JND is the same across the range. And that implies, well, if you're interested, I'll tell you what it implies. I'm not going to go into it otherwise. We looked at the effective frequency. There's no effective frequency on these JNDs. That is, if you present two stimuli at 50 hertz of different amplitudes, or two stimuli at 200 hertz of different amplitudes, their discriminability are going to be the same. Duration matters up to a point again. After 200 milliseconds, it basically plateaus. So here's the thing that we did that might be interesting to you, to some of you who are interested in doing this at home, or not at home. That was, yeah. Anyways, is a model. It's really been published for a couple of years now, but where we basically wanted to see if we could predict how detectable and discriminable a stimulus was using a simple model, so that others don't have to do this tedious work, these tedious experiments that we've done. You know, basically, ideally, if you had a new stimulus that we've never presented, and you want to see how detectable or it is, or how discriminable it is from another stimulus, you could plug it into this model, and it would tell you that. And so the model is super simple. Basically, we create a volume of integrated fire neurons. We stick a virtual electrode in the middle of that volume. This is an empirical function. The probability that you're going to excite a neuron in cortex is inversely proportional to the square of the distance between it and the electrode. So we build that into the model. We have an integration time window. Like I said, if you extend the stimulus beyond a certain time, it stops helping. That's what this is here to model. So then we model the population response to one stimulus, model the population response to another stimulus, and we have now these distribution of responses, and we can count the number of times the response to one stimulus was greater than the response to the other using ideal observer analysis, and that'll predict that'll be our prediction of the performance of the animal on that particular pair. Take-home message. All this is published in JND, so you can check it out. But take-home message is using this model, it has like 10 or so parameters, we can predict with 96% precision how detectable any stimulus will be, or how discriminable two stimuli will be from one another, right? Which sort of obviates the need of doing more of these super tedious experiments. If you have a new stimulation regime, you can plug it into this model, and it'll predict how detectable or discriminable it is. Okay. Now back to the encoding algorithms. I told you, like, DARPA wanted an encoding algorithm. They don't really care about the psychophysics. They just want you something that, again, take the output of the sensors, convert it into patterns of electrical stimulation of the brain. Okay. So how do we build an encoding algorithm from these behavioral results? Well, so we had data from each of these animals and each of these electrodes each of these animals for the mechanical pressure discrimination on how well they could stimulate applied to the skin. And then on the same day, the same animals and in matched locations, we had them do the exact same task with electrical stimuli applied to the brain. Oops. So we made the following assumption. If something is 80% detectable mechanically or 80% discriminable mechanically, and something is 80% detectable or discriminable electrically, they are perceptually equivalent, right? And so we can sort of match this mechanical amplitude with this electrical amplitude. And we can do this across the board at each discrimination level to get a function that looks like this. So this is electrical amplitude mapped on mechanical amplitude. Each of these curves correspond to different electrodes. Different colors correspond to different monkeys. Let's focus on Paps because he was weirdly consistent. This means that if you wanted to produce a sensation that was perceptually equivalent to a 0.5 millimeter indentation of the skin, you would need to electrically stimulate the brain at 60 microamps. So this is precisely the encoding algorithm that you need, right? That maps mechanical onto electrical, okay? So there's a lot of psychophysics, and maybe everything kind of got a little blurry, maybe you took a little nap. So this is the part of the talk where I prove to you that this function does what we claim it does. I should also point out that there's a lot of variability in Macallan, and there's some differences between PAPS and Macallan, but they all have this in common, that really they're sort of negatively accelerated functions, and the differences between all of these are relatively minor, right? And so, you could imagine, and this is an ongoing point of research in my lab, that just a single function could be used to approximate all these functions because of the general shape of it. Okay, but this is the proof in the pudding part of the talk. We devised this algorithm, which you may or may not understand why it should work, but then we tested it, okay? So what we did is we had the animals do this pressure discrimination task, but instead of delivering the stimuli to their skin, we delivered the stimuli to a prosthetic finger with a sensor, a pressure sensor. So this is what a trial would look like. You indent the pressure sensor with a weak stimulus, and you get this time-varying pressure output here. A stronger stimulus, this is the time-varying pressure output there, right? We take in real time the output of this pressure sensor, we pump it through our encoding algorithm, and we get out the electrical stimulus. An electrical stimulus that we purport should produce a sensation that is equivalent to this mechanical stimulus. And so you get a weaker electrical stimulus for the weak stimulus than for the strong stimulus. I've been talking about pressure, but I've been using units of amplitude, and you guys are engineers, so you probably know that, well, that doesn't really make sense. This is just to show you that depth of indentation into the skin, which is what I've been reporting as a gauge of pressure, is really monotonically related and linearly related over this range with pressure. So this is 75 micron indentation, 100 micron, 200, 350 micron indentations. This is the time varying pressure output, and you can see they're linearly related to one another. And of course, as the pressure increases, so does the electrical stimuli applied to the brain. So anyway, so the animals we're using this sensory encoding algorithm to see how well the animals could discriminate pressure based on this brain interface. And this is the data. So the purple trace here is, trace like what I showed you earlier for the low standard and the high standard. The two magenta traces are the animals doing the same pressure discrimination task, but now based on these signals through the brain interface. And as you can see, and as I told you they would, the performance is as good with through the prosthesis as it is with the skin. Right? And in fact, I could have made them even better. I just need to crank up the gain, and you can make the animals better than with the prosthesis than with the mechanical stimulus, or the stimulus applied to the skin. But what you gain in sensitivity, you lose in range, right? We've capped all stimulation at 100 microamps. You can maybe go higher, but we haven't found that out yet because we don't want to burn holes in the brain. So we go up to 100 microamps. So our range is basically 0 to 100 microamps. So what we want to do is maximize our range and make it as natural as possible. And this shows that within this naturalistic range, you can get several increments of discriminability. But anyways, the point is, though, that the point of this function is that our mapping function between electrical and mechanical does what we claim it does, which means it's a mapping between electrical and mechanical such that the electrically induced sensation is perceptually equivalent to the mechanical one along this continuum. The other thing we did with these animals is once we did this, we paired a mechanical stimulus that was 440 microns, which we had determined using our function should be equivalent to 50 microamps, with electrical stimuli that vary between 20 and 80 microamps. And as our function predicted, if the stimulus was less than 50 microamps, it was perceived as less intense than the mechanical stimulus, and vice versa for stimuli greater than 50 microamps. This was super important because this showed that the animals, this was the first time they were comparing electrical and mechanical along this intensity continuum, and they could do it immediately. So it's not like two completely different sensory continuum. Okay, so we did all these experiments, and most of what I showed you is published already. And then we did these extremely boring experiments where we chronically electrically stimulated the brain for 6 months, took out the brain and to see if we burned holes in the brain, and we didn't. And we sent those data to the FDA, and the FDA approved human trials of these electrical stimulation, of electrical stimulation. So then at the University of Pittsburgh, a team led by Mike Boninger, Rob Gahn, Jen Collinger, and Andy Schwartz, implanted a patient with an electrode array in motor cortex and an electrode array in somatosensory cortex. And then just kind of implemented the algorithms that I showed you earlier. So this was published in Science Translational Medicine a couple years ago. And this is Rob here. And this is the patient Nathan, who has these arrays. Actually, has 3 arrays, maybe 4 in fact, 2 in motor and 2 in somatosensory. And the nice thing about humans is you don't have to train them. You can ask them, where do you feel that? And that's what is happening here, where there's this mapping between. Actually, you know what? Let me step back for 1 second. In a human, how is it that this stuff that I showed you? How is this implemented in a human, right? So unlike the monkeys, the humans are tetraplegics, so we can't map out their receptive fields, but they have language. So the idea is you map an electrode, let's say electrode 35, you electrically stimulate through electrode 35, and you ask the patient, where did you feel that? The patient will now be able to report where they felt it. So let's say they feel it on the index finger. So now you can connect the sensor on the index fingertip to electrode 35. Anytime the index fingertip touches something, you electrically stimulate through electrode 35, and the patient will feel something on his or her index fingertip, right? How much you stimulate through electrode 35 is going to depend on the pressure exerted on the sensor, and we can create a sensory experience that is appropriate to that level of pressure by using our function, right? So here, this is just showing that when you do this mapping between electrodes and sensors properly, the patient just, when you touch a finger, he experiences it on that corresponding finger. And he gets it right every single time. And Rob, this is not shown in this video, but occasionally Rob actually would do 2 fingers at a time to see if the patient would report that, and he often got, just unbeknownst to the patient, was like, hey, that felt like it was my index and my ring finger. The other thing that they did a little bit of, but this is so boring, you don't want to subject tetraplegic patients to this extensively, but they did some of these detection experiments, discrimination experiments, and found very similar results that we did with our monkeys, which is nice because that means we can use this model that we devised for the monkey for the humans, and you don't have to now map out this whole sensory space all over again. So, but I started this talk talking about, it's great, we're doing touch and fantastic, but the important thing is, and what I framed this entire talk with is that this should make you better at using this hand, right? And the question is, does it make you better? And so this is a task, the same task I showed you earlier, but now this is with Nathan, with stimulation and without stimulation. And hopefully you can see just from looking at this that he's much better with stimulation and without. And I'll show you one more. And so this is what the movie is like. This is what the data looked like. So you have, with stimulation, you get a better score on this task than without stimulation. And this is just to show you that basically the reason for that is that you're actually faster doing it with stimulation than without, right? So your completion times are significantly faster. And then when you ask the subject, why is it that you're faster, is with one of the key points, and this is really early days on the sensory feedback, but at least based on self-reports, what makes the patient better is that they know when they've made contact. They don't have to rely on the visual system. Whereas when they're doing the task with just vision, they have to say, well, some of these fingers are occluded. It takes them a while to figure out that they made contact and that they can move the thing. And I'll just end with this slide here. So this is Jan, This is Nathan. And so not only has the sensory, helped to make these, but you can also see that Nathan is better at this, not because he's intrinsically better than Jan, but because these algorithms, the motor decoding algorithms have been improving steadily over time, and I think that kind of shows in these videos here. So I just want to quickly acknowledge the people in my lab, but you've already seen them in earlier slides that have done the work. And I also want to acknowledge my colleagues. So this was Revolutionizing Prosthetics was run out of Applied Physics Lab. Mike McLaughlin, who's now gone, was the PI. Revolutionizing Prosthetics is ending in a few months. It's one of the longest running DARPA programs, but it's going to end soon. And I had a close collaboration and ongoing collaboration with University of Pittsburgh. As I said, Mike Boninger, Collinger, Gaunt, and Schwartz were the main players there. And I just want to acknowledge my funding sources. Most of this was funded by DARPA. Thank you for your attention.
Sliman, great talk. So I'll give you a token of our appreciation.
Thanks.
So,
Do I open it now, or is it weird?
maybe later.
I'll just wait later.
I don't know if they have a microphone. Maybe not.
Yeah, thanks very much for coming. I have a question about the set. So you say that the mechanical is equal, equivalent to the electrical.
Great.
But is it really true?
No.
Blue is true in what you measure.
Yes.
But what about the patients? What do they?
Right, great. So that's a fantastic
Can I ask
question.
you to repeat the question?
So the question was, I used the word equivalent. The sensory experience, the mechanically induced sensory experience was equivalent to the electrically induced one along this continuum. That's exactly right. So it was along this intensive continuum, they were equivalent. Were they the same, indistinguishable? Absolutely not. And if I gave you that impression, I apologize, not at all the case. That being said, and with the monkeys, you have no idea. You can't ask them, how did that feel? Did that feel natural? With the humans, though, what was surprising with the humans, and these are flat trains, so electrical, turn on, you have a pulse train and you turn it off. They reported the evoked sensations as nearly natural, which at the time was kind of exciting. Since then, though, and that's what's in the paper, since then, though, I've been working with Dustin on naturalness in his amputee patients, and turns out naturalness is not a super reliable. So maybe we just lucked out with this patient using the word natural, right? And this patient, by the way, had been deafferented for 10 years, so it's not even clear that he would even know what a natural sensation would be anymore. So what I would say is the quality is definitely not the same as a natural touch, right? There's no chance that would be true. But what's also true is it's not unpleasant, right? So no pain was ever elicited, and subjects really don't report it as a weird paresthesia. So that's about as much as I'm willing to say about that.
Could the difference be in that we stimulate the peripheral nervous system and you're stimulating the cortex directly? So by the time the signal gets to the cortex, it's already been processed?
That's absolutely possible. There's all kind, it's hard for me to speculate. So I think the question you're asking is a difference between the sort of naturalism of nerve-induced activation to cortex-induced activation due something to the fact that in the nerve there's all kinds of processing that goes on and not in cortex. It could very well be the case, but let me put it this way. Dustin, Emily, and I are going to keep working at the nerve, on the nerve side, and I think there's certain things we can still try to make things more natural. But again, I don't even think that that's the important thing, right? The important thing is, it'd be great to say, oh my God, what a wonderful, velvet you have, with the prosthesis. But the first goal is not that. The first goal is to improve the dexterity. And I don't think naturalness necessarily is necessary. Naturalness is necessary to improve dexterity. And that's really the focus is on dexterity.
So your earlier kind of question about how much you can actually learn, how
Yes.
does that jive, that relevance jive with, if it's not natural, one would suspect that it's
Great.
It's almost a form of substitution, so you're kind of like,
Right.
where do you draw
Right.
those?
No, that's a beautiful question. So the question that we ask ourselves is, what are the key aspects of the neural response? Or are there aspects of the neural response that we can reproduce that are going to be more what the rest of the brain expects? Like, let me just be more specific than that. One of the things that we're working on right now is looking at the neural correlates of contact events. When you make contact, when you break contact, what does that look like, right? Everything I've shown you here is just like, in fact, I think I might have a slide on that, yep. Everything that I've showed you here is like flat trains, or we're just tracking pressure, right? And that is not at all, it turns out, how the brain encodes contact events, right? What happens is shown here, when you indent the skin, right? You have these monster transients at the onset and the offset of the indentation, and very, very weak, like 10-fold weaker activation during maintained contact, right? And this is true in the nerve, and this is true in the brain, right? So here's just a kind of reasoning that we would think is, if this is such a general principle of tactile coding along the somatosensory neuroaxis, one interpretation is that this is one of the keys, right, where you have these huge transients that give you very precise information about where and when contact happened, and that's right, and so we want to reproduce that. I think that's where we're at right now in our thinking, is we want to reproduce that as a first pass. And this is consistent with a theory that was put forth by Roland Johansson and Ben and Dean like 20 or so years ago, that really what the tactile system is there for is to mark transitions between subtasks, right? So okay, now I'm reaching. Oh, I've made contact, so my reach is done. Now I grasp. Oh, my grasp is done. You know what I mean? Now I can start lifting. And so that's what one of the fundamental goals of the somatosensory system, or these tactile signals are. So we want to reproduce that information and convey that information as naturalistically as possible. But I just want to say one last thing is, given the state of technology, there's no freaking way we're going to, in the near future or maybe ever, be able to reproduce the sense of touch in all its glory. So, but what we can hope to do is, the somatotopy, respecting the somatotopy is a no-brainer, no pun intended. You do that or you're dumb. But also the dynamics, I think, we see these dynamics are so consistent, so salient, that I think if we reproduce these dynamics, we'll have more useful signals. And in fact, there's some not yet convincing evidence that that's the case. Okay, there's too many here. Bob, here.
I have a very practical question. So you showed the areas for the digits and all. They're pretty precise. So how do you get your arrays to line up with
Sure.
the areas that you want to do? And I'm going to go back to the resolution issue.
Yeah.
Like, can you make it wider and have a better? There's a trade-off there. So you're using the Utah arrays.
Right.
Like if you were doing a new one, how would you change?
Right. So there's a bunch of questions there. Let me try to answer them if my memory will let me. So one question is, how do you know where to put the array? In monkeys, it's trivially easy and it's beautiful. Because monkeys, here's the central sulcus, here's the interparietal sulcus. Wherever this interparietal sulcus sort of curves towards the central sulcus, that's where the face is and that's where the hand is. And I put 30 of these in and I always hit hand and no problem. With humans, it's way trickier. I don't know if you guys, as evidenced by the latest paper by Richard Anderson, where you completely missed the hand representation. Although honestly, you've got to try to be that off. But with humans, it's way trickier. And we're actually currently trying to figure out how to do that. So even the PIT group did better in that they hit the hand, but they didn't hit the distal digits. And what we're working on right now is on an fMRI protocol, seeing if we, with even a 3T, but maybe with a 7T, identify specifically the digit representation relative to the proximal digit representations. I mean, the distal versus proximal digit representation, so we can put our array in just the right place. And no one has done that in humans, as far as I know. But we're working on it.
Can you comment, does your participant Nathan
Yes.
Get any sort of proprioceptive feedback?
No. It's a short answer. No, the reason I'm kind of being weird is that there's this paper that just came out in eLife by Richard Anderson, where he claimed that there were proprioceptive percepts were evoked, and we had a long conversation about it several times, in fact, over the last 24 hours, or basically over the last week. You would not expect proprioceptive sensations in area 1 and 3B. We targeted in all these studies the tactile representations. If you went into area 3A or 2, you might expect proprioceptive sensations. This is why it's a little bit surprising in this Anderson paper that they would get proprioceptive sensations through area 1's activation or stimulation. I'm also very skeptical that they actually got proprioceptive sensations. So
I should probably warn you that the Anderson Lab is one of the biggest viewers of our
That's fine. I'm just being honest.
Talk about what cortical layer you think you're activating.
Right. So we thought a little bit about this. In area 1, with our UEAs, they're 1.5 millimeter long, so they're all subgranular, right? In 3B though, and so we can't because they're roughly in the same layer, so it's hard to tell. In 3B though, we're straddling the bank, right? So some of these electrodes are supragranular, some of these electrodes are infragranular. And so we looked, is there, so you would expect a posterior to anterior gradient in threshold or something looking at these electrodes, and we found nothing, right? So there might be an effect. If there is an effect, it's a weak one. Others have looked at this, and there is a gradient. I think you get more sensitive as you go deeper. Like I think Kevin Otto did that. But these effects are pretty minimal. It's not like your threshold's like double or something, or half. So we didn't see anything like that. And in fact, the other thing we found is that our 3B thresholds and our area 1 thresholds were almost identical, just laying right on top of each other. That's why I didn't, notice in the detection and discrimination, I didn't show you area 1 and 3B separately because they were so similar. So again, suggesting that maybe depth doesn't matter that much here.
So curious about the product performance, like from the time you map it, you start working with them,
Yes.
to the time that they,
Yes.
they end the study. How often do you have to recalibrate, and how good do they improve, or are they pretty much the same day?
It's funny you would ask that. Why are you so obsessed with calibration, man? I'm just kidding. So here's the thing. First of all, one of the nice things about electrical stimulation, unlike the decoding stuff, is these UEAs just decay with time and get worse and worse and worse. And eventually, if you're lucky, after a few years, they just are completely useless for recording. But in our hands and in that of others, the stimulation thresholds really are super, super stable. Like for 3 years, they're super stable. What happens is the thresholds don't change at all until one day the entire array fails and nothing works, right? And that's what our experience has been. And you can imagine what's happening is, the signals that you're trying to record are tiny, like microvolts, right? Tiny signals, and therefore, if your array gets a little bit encapsulated, you just can't do it anymore. But with electrical, you're just pumping large currents through them, and they just blast through that encapsulation, and it doesn't seem to have a big effect.
One last question, Nolan. So your ICMS is basically recreating a passive touch paradigm, right?
Yes.
What would change if you were, how would you create an active touch paradigm? Right.
So we're working on that. But here's a very good question. Everything we do is like the animal sitting there. Like, is this applicable to any kind of active touch paradigm? Here's why I think yes, is because when we take all the ideas that were developed using passive paradigms and we apply them to this ARAT task, where the human subject is doing things, we see an improvement in performance, right? So it seems to work, but in my lab now, we're starting to create environments where monkeys are interacting with virtual objects. We're tracking their hands, but they interact with virtual objects and get tactile feedback from these virtual objects through ICMS, and then we'll be able to test that more.
All right, awesome.
Thanks, guys.